Welcome to the Masters of Beautiful Achievements podcast, where we share the insights from inspiring masters about how the use of physics, biology, and green chemistry is shaping our future. It's all you need to know about innovation and better business models. The podcast is hosted by Alexander Prince, founder of Scope Matters. For more inspiring episodes, visit the Master of Beautiful Achievements website. Today's episode is part of a special series where we look at microbes through a different lens. We will explore how life sciences, biotechnology, and environmental sciences are influencing how we look at our natural world. During this series, Alexander Princeton and Mark van Bemmel are curious to know what happens when we zoom out and look at microbes as important players in a much broader system. How does this different perspective influences on how we look at the role of microbes and viruses in the web of life? And what lessons can we draw from nature's logic into making our world more sustainable? Together with specialists in the field of virology, microbiology, and biochemistry, we go on a journey combining insights from various disciplines to learn about the essential questions we might have been overlooking. And who knows, we might discover one or two innovative applications along the way. This series is brought to you by Scope Matters, a systemic innovation agency from Rotterdam, the Netherlands. They accelerate business leaders through systemic innovation by merging ecological thinking with economic doing. Their system approach transforms and improves the business model of their clients by creating new opportunities, mitigating risks, and increasing cash flows, while at the same time eliminating waste and doing good. It is a space where systemic sustainable thinking meets circular innovation, because in the end all matter counts. Check out the website on www.scopematters.com. And Orvion, a young biotech company based in Stolwijk, the Netherlands. They study microorganisms and are developing their own DNA analyzing techniques, like for instance the Orvion U-Detect. They work on solutions to remove contaminants from ground, water or industrial processes more cheaply and permanently. Their services are available for water treatment, soil remediation, ecology and industry. Orvion believes nature can do a much better job. So please take a look on their website on www.orvion.nl. And a big thank you to our Patreon community who are monthly supporting this podcast so that we can bring you these beautiful stories. You can find the link below this podcast on how to join and to see and learn more about the benefits. And now it's time to start the conversation.
So in today's conversation, we will explore how physics of water is impacting biological life as we know it. My name is Alexander Princeton, and together with my co-host Mark van Bemmel, we'll explore the fascinating world of microbiology. As water is such an important aspect of life, we decided it's time to ask an expert to join us in this conversation. And today, our special guest is Gerald Pollack from the University of Washington. He's a professor of bioengineering, and his Pollack laboratory is dedicated to unravel the mysteries of water. So welcome, Jerry, and it is an honor to have you in the podcast.
It's my pleasure to be with you guys. As you know, I have a love for Holland, and so connecting with me is very special.
And Mark, you have a background in microbiology from a next generation sequencing perspective. I assume water is also a very important aspect in the work you're doing. And in what way?
Yeah. Oh, you were asking Mark.
Mark, yeah. And then
I thought you were asking me.
Mark. After that, you can comment, Jerry.
Actually, I studied environmental technology in Wageningen University, and studying environmental technology, you will run into microbes sooner or later. And I've always worked on and be fascinated by what microbes can do. And obviously there's a big connection with water because microbes need water to grow anyway. And water is a very important route for the transport of all kinds of things, also contaminants. So I've worked a lot on groundwater, but also on water in surface water quality. I'm really interested in what you have found, why water is so important for life. Yeah.
Okay. Well, I mean, I'm not sure if that's your opening for me to discourse on the subject, or maybe I should wait for specific questions. You tell me. You guys are in.
No, go for it, Jerry. What is your take
Okay,
on
go
that?
for it. So yeah, microbes, water microbes, very important. But what we found is that in the microbes, as in all other cells, the water is not what we think. We often think that the water inside those cells, the microbes, and in fact all cells in your body, we think that it's liquid water, but it's not liquid water. And the way you can determine that yourself is just merely to cut yourself while you're shaving, for example. You cut yourself. Now, if all the cells contain liquid water, once you cut yourself, the liquid water would come pouring out of your body, as it would in a fracture in a water-containing pipe or conduit. It comes pouring out, but it doesn't come pouring out. So it means that the water is maybe something different, and that's what we discovered. So I guess let me expound a little bit on what we found, because it really does impact every cell in your body, beyond the microbes, other cells as well. And so I start with a little background. This will take a few minutes, if that's okay. Yeah. So the background starts with a few famous scientists on whose shoulders our work stands. And our work involves a different phase of water that we discovered. You know, we all think that water has three phases. We learned this in middle school. We learned that water is a solid or liquid or vapor. But we discovered a fourth phase. And this fourth phase is a phase where the molecules are actually organized in some way, and I'll get to that in a moment. It's sort of like a crystal, or a liquid crystal, which has the consistency of like a gel, like for example, raw egg white, and that's why when you cut yourself, it doesn't come pouring out. It stays where it is. So who were these giants? The giants are, well, there are two of them. There are actually more, but there are a couple. And one of them is the famous Hungarian scientist. I guess you'd call him a biochemist. Many people refer to him as the father of modern biochemistry. That's Albert St. Georgi. And he won a Nobel Prize for discovering vitamin C. But as a really creative guy, he went on to study many phenomena, and he was, you might say, a Nobelist among Nobelists, really reaching the pinnacle. And he knew about water, that water was not just a background carrier of the more important molecules of life, you know, like the water in the bathtub in which you sit. Not like that at all, but the water is really important in whatever the cell does. And one of his famous aphorisms is life is water dancing to the tune of solids. So he knew that water was absolutely central for everything that cells do. That was Albert St. Georgi. And then came Gilbert Ling, and Gilbert, unfortunately, it passed just shy of 100 years old about a year ago. And Gilbert came from China. He was in the first cohort of young aspiring scientists chosen from all of China. So you can imagine they chose three of them, and they chose a physicist, a chemist, and a biologist. And he was the biologist, and the physicist went on to win a Nobel Prize. And so you can get some idea of the quality of these three people. And Gilbert made his mark in a, you might say, a very controversial way. He had evidence that inside the cell, that the water was not like water in a glass. The water molecules were organized like soldiers standing at attention. One molecule stuck to another, and they're all kind of lined up. And well, I met him more than 2 decades ago. I met him at a conference in Hungary, and I was really impressed. I hadn't known much about his work. By the time he passed, he had written, I think, 7 books on the subject, and almost everything had to do with the water, with evidence for the structure of water, which he maintained throughout his career was different from ordinary liquid water. And by the way, what I want to tell you is that what we found goes in the same direction as what Gilbert was talking about, but it's somewhat different. I'll make that clear in a moment. But anyway, Gilbert was arguing that the water in biology is different from the ordinary liquid water that we're talking about. So having met him at this Hungarian conference, and having heard from a dozen other people who had evidence to support Gilbert's point of view, I was really impressed. On the other hand, I was feeling a little uneasy. Could this guy really be right? Because this is so radical, such a radical departure from what's in the ordinary textbook. So I gave one of his books to some of my students, and the response was uniform excitement. They said, if this guy is right, all of biology is wrong, because you gotta start all over again, because this is just so different. And so that kind of reinforced my own sense that these guys were right, that this guy is onto something that's really important. So I'm thinking, well, what are we going to do about this? Because I can't just sit on my rear end and say, well, yeah, good idea. I'm not that kind of person. So first thing I thought, well, I got to write a book, because Gilbert Ling had already written several books, but it takes a genius to read through and really understand what the guy is talking about. And I think it's partly it's partly because I can surmise he would sit down at the word processor, or before that at the typewriter, bat something out and send it to the publisher. You can't do that. Everything that's written requires editing. And the more editing you do, the clearer it becomes. And I think Gilbert didn't subscribe to that particular point of view. So it's a real challenge, even for geniuses like you guys, try to get through. But so I thought, okay, well, I think I can write things a bit more clearly. And so in putting out the book that I actually put out, it's called Cells, Gels, and the Engines of Life, from about 20 years ago. And 2 goals. One is to make clear Gilbert's contributions, and the second was to go beyond what Gilbert was talking about, and demonstrate exactly how the cell itself exploits this kind of he called it structured water. We now call it 4th phase, or EZ water, exclusion zone water, but anyway, that's not centrally germane. So I did all of that in the book, and I described how the evidence for what cells do with that water. And so if the cell is, for example, in its quiescent state, for example, let's say a muscle cell that's not contracting, then the water is the so-called structured water that Gilbert was talking about. But once the cell transitions into action, and for the muscle cell, that would be that the muscle begins to contract. Everything changes. The proteins change, and the water changes. The water changes from what Gilbert was talking about structured water to ordinary water, and then back again after the contraction is over. And for a nerve cell, it would be that if the nerve is not conducting no information, the water is structured, and together with the information transmission is the water undergoes a transition. So anyway, those were the two themes of that book, and the book was received with mixed reaction, shall we say. I see the smiles on your faces because you know. So reaction, some people wrote, oh, this is more of that crazy guy Gilbert Ling. Pay no attention because we know that Gilbert Ling is a crackpot. You know, reflexive criticism from those who don't appreciate hearing a different
voice. Yeah.
Yeah.
You know very well. But on the other hand, there were some others like well-known cell biologist from Harvard University. Harvard must be right because he comes from Harvard. He said, this is a 304-page preface to the future of cell biology. I like that one. That was a better one. Anyway, so I wrote this book, and some people appreciated it, others did not appreciate it. And it's got pretty pictures. Anyway, so I then thought we've got to plunge into this and do some experimental work to follow up because it's critically important. And of course, there was no money to follow up, so I sort of borrowed money, so to speak, from money granted to study muscle contraction, which was our field, and used that money to start studying water. And that was, I guess, close to 20 years ago. And within 1 year, we had remarkable results. And so I'll just conclude this long diatribe by telling you the essence of what we found, and therefore what is this fourth phase of water? What's it all about? Because it is central for everything in biology and beyond.
And the way you're explaining it, I mean, it's very similar to like how membranes work. So it's like a gel-like structure. I mean, we had in a previous conversation we had the guy from TU Delft, Mark van Loosdrecht, the professor, and he's into wastewater treatment, but also in biofilms.
Yeah.
And the way you're describing it, it's sort of like I don't know if it's a biofilm, but it's like a film-like structure where interesting things happen on a nano level.
Well, you might, yeah, you might put it in those terms, but this is more than a nano level. This kind of water that I'm talking about, and it will become clear in a moment, I hope, it can extend over appreciable distances, not just on a nano level, but a micro level, and even a millimeter level and beyond.
Oh, wow.
So there's a lot of this. This is more than just a laboratory curiosity. There's a whole lot of it. It's just that except for a few scientists in the past, it hasn't really been widely recognized. But I think we now have the evidence for it. So let me just tell you what we did and what it and a few of the major implications, and we could then build from there. So what we took some water, and we put little particles in the water. They're called microspheres, little tiny spheres, just representative particles. Doesn't really matter what the particles are because we've used many particles and the result is the same. And so you put the particles in the water, and then you immerse some material in the water, like a gel, for example. And we started with gels. So you take a gel, just like the gel that you might have for dessert, and immerse it in the water, and then see what happens. And what happened, we look in the microscope to see, but you actually don't need a microscope. Even a magnifying glass will do, and even your eyes are in some cases enough to see it. What happens is that all of those little particles start migrating away from the surface of the gel, and they leave they leave a microsphere-free region adjacent to the gel. And this region can grow, and as I was alluding to before, it can grow up to a millimeter in size under the right conditions, and we've even seen in some extreme conditions up to a meter.
Oh wow!
Yeah, this is we published that. Those are extreme conditions, but okay. So the first thing is there's a region that expels the microspheres, and it can be pretty large. And this region we call exclusion zone because it excludes. Someone suggested that. Right. Sort of no-brainer. But actually, and it really works well, not in Holland, but in the US, because exclusion zone, we say EZ, and it's easy to remember. So, but unfortunately in Europe it's EZ, and so it doesn't quite work as well when you say it's EZ to remember. Anyway, EZ, exclusion zone, or later, and I think more appropriately, we called it fourth phase water. This region that pushes out all of the particles, and later we found it pushes out basically almost everything. So, okay, so that's what we found. Next thing we found, we stuck electrodes in it, tiny electrodes called microelectrodes. In fact, invented by the same Gilbert Ling, who should have gotten 2 Nobel Prizes, one for that because that technique is so widely used, and also for the idea about the water. Anyway, he didn't. Okay. So we use those electrodes, and we expected, since water is neutral, we expected that this EZ region, this microsphere-free region, would be neutral. We found it wasn't. We found it had negative charge. And at the same time, we found that using a different technique, that the region beyond that had positive charge, equal positive charge. So what's going on there? Well, what's going on, we found that the water molecules are splitting. You know, water is H2O, that is H plus, OH minus. And if you can split the H2O, you get H plus and you get OH minus. And those OH minuses were building this exclusion zone. And the H pluses, nowhere to go, they get cast out into the water beyond the exclusion zone. So you have something like a battery with the exclusion zone being negative and the region beyond being positive.
Makes me curious, what would be the pH level then?
Of a pH level of what?
Of the exclusion zone.
Well, you know, pH is measured for a liquid. So, you know, you can't really define a pH of something that's, you know, it's a semi-solid.
Okay.
But if you were to break it up, you'd find, since it has net negative charge and it's full of OH minuses, you should be able to measure actually a high pH as a result. And the region beyond is a low pH. That's where the positive charge are. And that we've confirmed using pH-sensitive dyes and other techniques. So the main point, though, is that you got a battery because you got minus and plus separated, just like a battery. And we demonstrated that if you put electrodes in, one in the negative region, one in the positive region, you can light a light bulb.
Really?
So, yeah, this comes from water. But if you're thinking, which you guys do all the time, I know, you say, well, wait a second, my cell phone battery needs to get charged. This is a battery. It's got to get charged. And what charges the battery, right?
Sure.
Electrons, yeah.
It also costs energy to build the EZ, I would say. Yeah.
Yeah, exactly. So, you guys are, you know, you're right on. But where do the electrons come from? Is, you know, and we couldn't figure it out. We spent a few years scratching our collective heads. That's why my hair is missing. Too much head scratching. Where on earth could this energy come from?
But then you need protons, or.
Well, you need energy. Okay, and then how the energy works could tell you where yeah, you can throw electrons in and throw protons in, but they would recombine instantly. So you need something that keeps those protons out of the fourth phase or out of the easy. Yeah,
driving
but
force.
where is the energy? Where on Earth? So well, we found out, and it was a young, we say undergraduate student, 18, 19 years old, and we often have a lot of those students working in a laboratory, and this guy was doing what he was not supposed to do. So he's working on the laboratory bench, and he noticed right next to him was a lamp, one of these gooseneck lamps. You can bend it, and he shined it on the chamber that he was working with that had the gel and the exclusion zone and such. And he noticed that where the light was shining, the exclusion zone grew, and it grew wildly. I mean, by a huge amount. And so he called me in, and I was astonished. Of course, I thought, oh my goodness, this guy has the answer to the question: where does the energy come from? It looked like it was coming from light. And I said to him, well, why don't you remove the lamp and see what happens? And so he removed the lamp, and that the exclusion zone, which had grown, went back to its original size and such. So I was, of course, I was impressed. The student, I think, was sort of unaware of the import of what he had discovered inadvertently. And then we did real experiments, and we wanted to know which wavelengths of light were the most relevant. So we studied a range of wavelengths, starting with the ultraviolet, getting longer wavelengths into the visible spectrum, and then finally into the infrared. And we found that most of those wavelengths were irrelevant. They did nothing in terms of EZ growth. But the infrared, that was the one that really mattered. It was wildly different from the other wavelengths. And we could, by shining even weak infrared for tens of minutes, very weak from a light-emitting diode LED, we could grow the exclusion zone to 10 times the original size.
Did it grow in thickness or in
thickness?
I guess it's on flat on the surface, or
well,
yeah.
So we found later. I explained it grows in layers. So you start with the gel surface, and the first layer is a molecular layer that's different from what Gilbert Ling was talking about. It actually is a honeycomb. It actually consists of an array of hexagons consisting of oxygens and hydrogens, and that layer then acts as a template for the growth of the next layer, and the next, and the next. So these layers, these hexagonal honeycomb layers, keep growing one by one. And so if you shine more light, you get more layers. As simple as that. If you remove the light, then these layers disintegrate, and you get
thicker, and it also extends more.
Well, it basically gets thicker because you got more layers.
Right.
In terms of the extensiveness, we didn't study that because usually if you have a gel, then these layers will occupy the full surface of the gel, and so. That's a matter. The extensiveness is a matter of the extensiveness of the nucleating surface.
So how do you measure the thickness of this?
You just look in a microscope, and you can see
then
it.
You look at the spheres that are pushed out.
Right. You just look at where there aren't any spheres, and the demarcation line is very clear.
And are these spheres charged, then also negatively charged, so that they are pushed out by charge, or?
We've used different spheres and different materials that are negatively charged, neutral, or even positively charged. They're all pushed out. So it's not an electrostatic kind of although charges are certainly involved, but it's that these sheets, these hexagonal sheets, are so densely packed that essentially nothing can get in, and they're pure. So everything, any kind of contaminants, if you will, like particles or molecules, get pushed out. Just like when ice forms, the ice is a pure crystal, and if you start with water that's not so pure, in order for the ice crystals to form, they need to push out these impurities. And that's why, for example, at the bottom of a glacier, you have all these impurities, the glacial moraine sitting at the tongue of the glacier, sticking out. The principle is the same. So, okay, so we got that. I'm trying to think. If those are the major features that.
But that is weird in a way that water. I mean, I know always forget the name of the professor in the UK, but he had a website stating the 77 anomalies of water.
Oh yeah, sure, yeah.
And I mean, it's the most widely available molecule, so it has to have a very important function to do. And the way you're describing it is sort of like a cleanching effect of water. I mean, if it doesn't want to have all the other stuff, it just makes sure it cleans itself. But then that's where I want to go. Why ultraviolet light? Why would it grow on that? I mean, what's in there that it stimulates?
Good question, and we've just been addressing that. But before I answer that question, may I just go back to those anomalies that you're talking about? And if I forget the question, please remind me. I have a tendency to do that. Yeah, those famous anomalies, and the anomalies are based on the presumption that water has three phases, right? A solid, liquid, vapor. But if there's a fourth phase, then those anomalies, well, I should say that many of those anomalies resolve right away with this fourth phase. That's actually one of the motivating issues. There are so many anomalies. In fact, anomalies were known for 100 years, and that's what prompted physical chemists even 100 years ago to say, something's wrong here. There are too many anomalies. Even then, 100 years ago, there were so many that the idea is, well, there's got to be something is wrong with our assumption that there are three phases. There's got to be something else. Okay, so now to repeat your question, what is it about the surface that really matters in terms of growing this easy? Was that your question?
Yeah, and why infrared?
Okay, so we got two questions. So, again, remind me of the second if I finish the first, or when I finish the first. Okay. So first of all, not all surfaces nucleate the growth of EZ. For example, if you have a hydrophobic surface like Teflon, for example, where the water is resting, it beads up, no such phenomenon happens. It's only when you have hydrophilic surfaces, water-loving surfaces, so a surface on which if you were to drop some water, it spreads out. The surface loves the water, hydrophilic, water-loving surface. But not every surface does that. We found that, I should say, most surfaces that we've looked at do that, especially gels. If you have a gel-like surface that has some compliance to it, then we get it consistently. On the other hand, if you have a hard surface that lacks such compliance, and then some surfaces do, some surfaces don't. And we found out through some experiments that if you take a given surface that has compliance to it, and it builds, if you put cross-linkers between the molecules to make it stiffer, it gradually becomes unable to nucleate this. And so we come to the conclusion that it's probably the distribution of charges on this nucleating surface that matters. You see, the charges need to match the charges on the first layer of EZ water. And those charges are very regularly arrayed. I mentioned to you that it's a honeycomb structure full of hexagons, and those charges are at very specific points. And so if those charges match the charges on the nucleating surface, then you get growth. If not, you don't get growth, or you get limited growth. So that's how we think it works, and that's why most hydrophilic surfaces with just a little bit of compliance can nucleate the growth, but hydrophobic surfaces don't do it. Okay.
And
So
why infrared?
Okay, why infrared? The answer is we don't know. However, we surmise that when infrared energy hits the water molecules, it breaks the water molecules into negative and positive parts. It starts wiggling the molecules around, and so the OH minus and the H plus separates and makes those OH minuses able to glom onto the surface. I'm simplifying something that's maybe a bit more complicated, but we don't know the answer. This is only a conjecture. It's what we surmise. And infrared energy is all around us.
Yeah.
If you were to take infrared camera in complete darkness, in other words, the sensor in the camera is not sensitive to visible light, but to infrared light. You get a beautiful image, even in complete darkness, and which proves that infrared energy is being generated by everything around us. So it's abundant, which means that EZ water is abundant. It's all over, because the energy that's required for building it is likewise all over. And if you increase the energy, you get more EZ. And if you return to the default state, it goes back to the default amount of EZ. Sorry.
Then it would build everywhere where there's a suitable surface and enough water. Then.
Exactly. Yeah.
Right.
Yeah, and that's almost everywhere. And it also builds around even molecules. For example, proteins inside the cell. They all have a layer, it's well known, of so-called interfacial water, and we understand now that interfacial water we designate to be EZ water.
Right. Proteins inside the cell, also on the cell membranes. Do you know?
Yeah, well, cell membrane also is there, but the cell membrane is only a small fraction of the cell, and this water is everywhere inside the cell, because the cell is filled with these molecules, proteins, nucleic acids. Proteins are like 70% of the solids inside the cell. And every proteins have charges on their surface. And all these charges nucleate the growth of EZ. So it means that essentially all the surfaces, the solid surfaces inside the cell, have EZ water around them. And because these solids are so densely packed inside the cell, it means that essentially all the water inside the cell is EZ water.
Right.
And that's why if you stick an electrode in the cell, you measure negative charge. It's not because of the membrane and the gadgetry inside the membrane. It's because the water that's in the cell has negative charge. And by the way, it sounds as though I'm just coming up with that statement off the cuff, but there are genuine arguments. I wrote a paper on that to dispel the notion that the electrical potential inside the cell is due to channels and pumps. I published that a few years ago, and just one reason is that if instead of looking at the cell, if you look at a gel, the inside of the cell is pretty much like a gel, but if you take a gel lacking any membrane and membrane gadgetry, the electrical potential in the gel, compared to outside, is the same as the electrical potential in the cell. You see, so this is a problem, because if you say in the cell, the electrical potential is due to some membrane gadgetry, then how do you explain the same electrical potential inside the gel, which has no such gadgetry or no such membrane?
So that brings me directly to the mitochondria. I mean, the engine within cells. It keeps tossing about in my mind. I mean, in a way, you're saying it might be powered by EZ water.
Well, yeah. I mean, this is a really good question that you raise. The question is, where do we get our energy? And And we think about mitochondria and the production of ATP, adenosine triphosphate, and we've all learned that the source of energy, it comes from metabolism, and from the ATP with its high energy phosphate bond. However, it's not as settled as we might think. And again, I refer back to Gilbert Ling. And if you look on his website, gilbertling.org, I think it's still in existence, you'll find in big bold letters the question about ATP. So Ling tells the story that I think about 70 years ago or even longer, a group discovered that there's a high energy phosphate bond in the ATP, and that's where the energy comes from. And that was quickly accepted because everybody was looking, where does our energy come from?
Right.
Yeah, and Gilbert points out that 1 year later, a group of chemists published a paper saying that the previous guys who said that there's a high energy bond made a mistake in their arithmetic. There is no high energy bond. They made a mistake. And Gilbert points out that since that time, nobody has ever addressed that issue or resolved it. So it remains unclear, was the original group correct in designating a high energy bond, or were they wrong? And there's no such high energy bond, you see. And so the question remains open. And even though there have been several generations in between, we actually don't know whether it's the high energy bond in the ATP that is responsible for all of the energy that we have, or something else. So that something else could be what I've been talking about, because those charges or separation of charges represent potential energy. It's like a battery. And the question is, does this battery operate inside our bodies? Well, it should operate. If physics works inside our body, it should play some role. And we've in my more recent book, The Fourth Phase of Water, I begin to outline that possible role. So we don't know whether the electrical energy that we have inside of us plays a minor role, a moderate role, or even potentially a complete role in generating the energy. So you guys are...
I'm extremely interested in degradation of difficult compounds, things like chlorinated solvents, but also now the PFAS molecules that are highly fluorinated big structures. I'm not sure if you heard about those molecules.
I'm not aware. Sorry.
Right, but often it is stated that these molecules cannot be cracked by bacteria, but in the end, often we find that there are ways for bacteria to do it. So they are able to use them as an electron acceptor, for instance, or as an electron donor in some cases. So could this mechanism or this buildup of electrons actually in the cell, would this help to get these kind of transformations of molecules?
I'm not sure, because I'm not familiar with the subject at hand, but these charges that are inside the cell clearly represent potential energy, because all electrons or negative charges want to get away from each other as far as possible, but they're all crowded together, so it's got lots of potential. And in the mechanism that I describe in the two books that I've mentioned, I described how that electrical energy that we're talking about, potential energy, can be used to drive the folding of proteins. Right. And the folding of proteins represents the work of the cell. It's what cells do. That's how they operate. And so there could be a direct link.
But for proteins and enzymes, is that the same, Mark?
Yeah. So So this electrical energy, you know, we simply don't know how much energy it's supplying to the body, but it can't be none because it's there, you know, and so it makes me think.
This electrical energy is within the cell. It helps for proteins to function, and it is driven by infrared light.
Yep.
So what happens if you have a good functioning organism and take out the infrared light? Does it stop functioning then, or?
You can't take out the infrared light because in your body, first of all, I guess I think your body is like everybody else's body.
I think so. Yes, I hope so.
I haven't examined it, but, I'm not sure, you know. But you've got metabolism going on. Metabolism generates heat, and heat is essentially similar to infrared energy. So you've got infrared energy from inside the core of your body, and you've also got it coming from outside, because I just mentioned that you've got infrared energy being generated by everything around you, and you're receiving it. And so you can't get rid of that energy, but the reverse is important, because if you add more infrared energy, that builds easy water inside your cells, and you feel better. And you can reverse dysfunction that way. And I think this is really important.
But if I am a bacteria living 50 meters below in deep soil, is there infrared light as well? I don't know.
Yeah, there's infrared energy. I mean, for example, infrared energy. Well, first of all, half of the sun's energy roughly is infrared energy, is heat that we feel. And then infrared energy is getting radiated from the core of the Earth outward at nighttime.
So it gets better.
Still coming from this way and coming from this way, from the bottom and from the top. You can't get rid of it. You see, it's always there, and so we're fortunate, right? That energy keeps coming in. We don't have to pay for it. You know, it's not like oil.
But you can put more on it, on a cell, and see what happens, is it?
Well, yeah, you can.
Yeah.
And you have the experience, maybe. Have you ever exposed yourself to the heat of a sauna, or as the Finns say, a sauna?
Yeah. And I like that. But
yeah, you feel better, right?
Yeah,
So why do you feel better? Well, I think the reason you feel better is that infrared energy is coming from outside, penetrating all the cells in your body, building easy water, and the cells in your body, whether it's your muscles or your brain or whatever, they come back to normal. And
it's like a cleaning effect.
Yeah, sure. So and that's also why people in Scandinavia, maybe some of the more clever Dutch people, will in the wintertime go south to feel the sun, exposed to the sun more, and feel better.
I used to grow up in the sun, Jerry, and when I ended up in the Netherlands because my parents decided it was time to get a proper education. But I was 8, and ever since I hate being in the northern hemisphere, because it gets dark in the winter, and it's something I still cannot cope with.
You've grown up farther south. Where did you grow up?
Venezuela and Oman.
My goodness. Okay.
I grew up around the equator, and
you still miss it.
I definitely still miss it. That's why the reason why we go in the wintertime for 2 weeks to Portugal, just to get back to the sun.
The good news in the northern hemisphere is that you have more of the sun in the summertime, and you have less of it in wintertime. So at the equator, it's more spread. It's even.
Yeah, I mean, were your parents correct in that you got a supposedly superior education? I'm not sure.
One thing
worthwhile, as many people presume. Go ahead.
Yeah, one thing I've learned about my world travels between 2013 and 2015, meeting similar minds like you, Jerry, is that it's not the education that matters; it's the way you look at the world which matters.
I think more and more of that, because some of the things that we learned from the textbook are not correct.
Mm.
And so, and we're rewarded for memorizing that often misinformation. It's drummed into us. And, you know, we get rewards like good grades for our ability to memorize and spout back that information, which in many cases is can be misinformation. You know, if I'm allowed,
Sure.
I think nature, Mother Nature, works in a simple way. I'm sorry for that telephone,
Never mind.
but I can't do anything about it. But it will stop after three rings, four rings. You know, Mother Nature works simply, and when we read something in the textbook, and we have to read it 3 times in order to begin to understand it, and we have to scratch our heads. What do they mean by this? How does this happen? Why does this happen? We're confused. It's not simple. We wonder about it. And I used to think, well, it must be some deficiency on my part. My gray cells are not as gray as they ought to be, or something like this. And I've come to realize that, well, that might indeed be correct, true. Deficiency in here, it may equally often be true that the idea that's being presented is simply erroneous. It's wrong, and the reason it's complicated to understand is not correct. And when I read something in the textbook, my first reaction is, if it's clear and simple, you know, intuitively obvious, probably it's right. If it's not, there's a good chance that it's wrong. And this is an area that's fertile for people to. You're laughing. Why are you laughing?
I had a hard time in university.
Yeah. You were.
No, I mean, I'm dyslexic, and all the professors writing their own syllabuses, most of them were also dyslexic, so that doesn't work very well. But I found it hard to understand certain parts of the theory, and eventually when I met people like you, I figured out that physics, biology, and chemistry can be very simple if you understand the principle. If you can explain the principles very easily, then the concept stands.
Yeah.
And nobody would ever able to explain them in easy terms, but keep it complicated. And then I'm just like, okay, whatever.
Yeah, you know, it's a challenge for the professor also, because you know, it's easy as a professor. I can tell you, it's easiest. You stand up in front of the class and just stop what the textbook says. Differently, you know, you really have to be on top of the material and be able to answer the questions that the students will inevitably ask. So
there is one question which was on the mind before we met. I understood from Robert Malone's newsletter that Luc Montagnier has passed away.
Unfortunately.
We were eager to get him on the podcast, but unfortunately that cannot be possible anymore. But I do know that he has also been intrigued on water and biological life. And I understood that he also looked into the 4th phase of water.
Well, yeah, I mean, he didn't look into the 4th phase exactly. But he was well aware. We were friends, and we spent a fair amount of time together. And his experimental results, many of them, but the most critical one, the most interesting one, is just astounding. And I if I may, I just reiterate.
Yeah. Interesting.
You know, if true, this is amazing, and apparently it's been duplicated by other laboratories. So he took some DNA suspended in a solution, and in a vial that was sealed, so nothing could come out, and put another vial nearby. And this vial contained just water, again, totally sealed. So there's no possible chemical communication between these two. He said the two are communicating through some either electromagnetic or other means, some kind of energy, some subtle energy. And the two are sitting beside one another for 24 hours, bombarded by some kind of generic energy like 50 hertz or 60 hertz or something like that to pump it up, so to speak. And then he removed this one, and he took this water, which he said had been informed by information coming from the DNA or from the water surrounding the DNA. He took this and created more DNA using the PCR method, that is now used every day for checking COVID.
We know it very well in our laboratory. Yeah,
we know it very well. Okay, and the new DNA that was created from the water had the same sequence as the original DNA. Imagine that. So it means that somehow the water sitting here got the information from the DNA, and that information was able to be used to produce more DNA, and that DNA had the same sequence as the original DNA. And so what that demonstrates is that there's some kind of energy, information, shall we say, information coming from the DNA that is somehow stored in the water. Otherwise, this could never have happened. This water must be somehow storing the information. So this is mind-bending because it means that that your dyslexic DNA is being transformed to others around you. And there have been lots of, it's not just the late Luc Montagnier, but other people who have demonstrated this. And, we organize conferences each year on the physics, chemistry, and biology of water. And now they're in Germany annually, and Luc Montagnier would come to each of these meetings and present the latest evidence for all of this stuff. But other people would present as well, different kinds of experiments that would demonstrate, not quite as dramatically as his, but clearly that water is capable of storing information. Now, it's not liquid water that can store the information, because if you think about the structure of ordinary liquid water, the molecules are randomly oriented, bouncing around at a furious number of times each femtosecond, and there's no obvious capability of storing information. And that concept, that inability, led to ultimately the demise of another famous French scientist who was actually a friend of Luc Montagnier, and that's how Luc got his start in this field. That was Jacques Benveniste. And Jacques demonstrated very clearly the existence of water memory. But at the time, nobody knew about EZ water and such, and so the world surrounding Jacques and his finding was simply unable to accept this as even a possible reality. And his career took a nosedive because of that, and because of what seemed like a vendetta against him from the editor of Nature and a committee of people, supposed peers, to come to his laboratory and look over his shoulder. And the peers included a famous magician who could figure out the tricks of other magicians. His name is James Randi, among others. And they reported that they went to visit the lab, and they reported these guys are doing sloppy science, not properly recorded, et cetera, and this is a trick. Well, they couldn't figure out the trick, and and the experiments have now been repeated by multiple groups and confirmed, and so water memory at the time dismissed because nobody knew about EZ water, which has the capability of storing information. All they knew is liquid water, and there's no such possibility. Forget it. Benveniste must be a trickster of some sort.
So is it sort of that the EZ water forms patterns, or what do you think?
It's not. Yeah, you might say it's patterns. It's there are two criteria for memory, and you can understand the criteria by looking at your laptop computer. the thumb drive that contains a memory that you can stick into your computer. So what are the characteristics? Number one, it's built of an array of transistors, and these transistors are regularly arrayed in a two-dimensional pattern, very regular, here, etc. And second, that each one of those entities has two states.
Semiconductors, right?
Yeah, based on semiconductors, either we say 1, 0, or on, off, or whatever, two states. And now look at easy water. It's got those two characteristics. The easy water, as I mentioned to you, these hexagonal honeycomb arrays are regular. They're geometric patterns, regular, just like the transistors. And the second is that they consist of oxygens and hydrogens, and the oxygens can have not 2 but 5 different states. So the typical so-called oxidation state is -2. It's what we learn in school. But there's also the same chemistry books that tell you that will tell you there's also -1, 0, +1, +2. So it means yes, that easy water has the very characteristics that are necessary to have memory. They have a regular structure, and one of the elements of that regular structure, the oxygen, can have different states. You see,
based on electrons.
Yeah, you might based on electrons. Yeah. So in the future, if you think about it, it's possible in the future that thumb drive that you have could be reduced to the size of a pinhead, a tiny pinhead, because the elements that we're talking about, they're on the atomic scale, not on the transistor scale.
Yeah, right.
We calculated, at least in theory, that could be reduced to something like a billion times the size that exists currently.
Yeah, well, we know the density of information of DNA, which is just immense already.
Right.
So,
exactly.
I mean, if you store all the world's information in DNA, all the world's information that's on disks and drives everywhere, then you need only 1 kilogram of DNA.
Is that what I did?
Right. Yeah.
That is ridiculous.
So nature has figured out how to
right.
do all
So
this.
it's all about information and how to read it, how to use it, how to transfer it, how to store it.
Well,
that's
Mark,
the challenge. That really is the challenge. But what it means for you and for both of you and for me is that we're full of easy water, and therefore if I'm conveying some sort of, shall we say, subtle information or biofield or whatever to you, you can get that information from me. I don't have to use my mouth to speak to you. I can convey that information. It's been known, of course, for how many thousands of years that this is the case, that we can exchange a nonverbal kind of information to one another, and people have demonstrated that this kind of information actually can be sent from one side of the earth to the other. Amazing, which suggests
cognitive resonance, is it called like that?
Entanglement, this quantum physics.
Entanglement, there are a lot of things that are attributed to quantum entanglement. It may or may not be attributed to that, but somehow the information can be transmitted. Transferred. Right.
But Mark, you're doing qPCR tests. I mean, with your nanopures and doing mostly bacterial analysis.
Right.
But what Gerald Jerry is saying that this is impacting your research.
Yeah, I don't know. Maybe somehow I have to give it a thought. I don't know.
Yeah,
I think nobody yet knows exactly how these things work in nature.
No, but this is.
We look at DNA molecules, and we try to read the information to understand it, and to put it into practice in a way. But I'm quite sure we don't know. There's so many things we don't know yet, or don't
No,
fully.
and DNA, remember, is surrounded by water, and
Yeah.
there's easy water that surrounds all biological molecules, including DNA, and the information could be also in the water that is
Right.
surrounding DNA. The DNA acting as a template, and then
Right.
you can kind of think about mutations that occur as some sort of
What we know is very interesting now that you mention mutations, is that mutations are always thought of to be completely random, but actually they are not. They are often on the same spots on the chromosomes, or it's not like that they are completely random and they take place everywhere. No,
and even in a
mutations occur at the same place and never at another space at another place. This is very.
I'm not aware of that. That's so interesting.
Yeah. And that research also states that these mutations have a memory of billions of years of evolution. That it mutates because it knows that a certain mutation is needed to solve a sort of a challenge at hand.
Well, I mean, would be one interpretation. Another interpretation is, that the mutation can occur because of some subtle signal that comes into the easy water, and then it's transferred to the DNA, and the DNA is more stable, and so more of a stable information storage site, and so it retains for billions of years or whatever. Yeah.
Yeah. And if
we know now much more. I mean, like 10 or 20 years ago, the DNA was thought to be for 95% or even more. It was thought to be junk or just random information from old stuff. But this doesn't appear to be true at all. I mean, there are epigenetic mechanisms that either put things on or off from the DNA molecule. So part of the DNA is used during very early stage growth, when you are an embryo in your mother's belly, and then that part of the DNA is used, and in a later stage another part of your DNA is used. So there are epigenetic mechanisms that use that sort of how do you say, regulate what part of your DNA is actually used at what stage of your life.
Oh, interesting. Yeah.
Which is so interesting. Yeah, maybe your easy water has some role in that kind of.
Well, it could easily.
I don't know.
It could easily play a role. Yeah.
But what we know is that the cell is an extremely complex machinery, and that everything functions, has its function and its role, and we don't yet understand. We understand only very little of it, I would say.
Well, yeah, and I think it all boils down to the foundation. If you try to build an edifice on a shaky foundation,
then it won't work.
You need all kinds of rubber bands to hold it together and such. But if you're really able to identify the true foundation, everything should rise from that foundation elegantly and neatly and simply. Yeah. And so.
I can give one. Example. If you have salamanders and lizards, if you pull off their tail, then it grows back again.
Yep.
And it is not many people know it, but when a very young child loses a part of one of his limbs, then it can also grow back.
Yep.
This is one of those epigenetic things that can be turned on or off in the DNA, and that's extremely interesting that nature has these kind of mechanisms for restoration.
I had a student who told me about his experience a few years. He was working with a saw and he accidentally cut off a finger.
Right.
He was able to rescue the finger, but he didn't know what to do, and so he called emergency services, and they recommended that there's a certain clinic that deals with this sort of thing, and they told him, "Oh, it's Friday afternoon. Why don't you come back early next week?" And he said, "What are you talking about? This is an emergency." They said, don't worry about it. Just put some gel on
Ah,
on the wound, and
nice.
come back in early next week, and we'll take care of it. And the finger grew back.
Wow.
So and this guy was not a baby. He was,
Yeah.
he was like 20 years old or something like that. So yeah, that epigenetics very important,
Yeah.
and yeah, and not only tails but fingers can grow back too.
Right.
It is very nice. Yeah.
I would really love to know a bit more about these easy or short phase water and DNA interactions. But I'm quite sure that it's an extremely interesting subject for research.
Oh, it's extremely interesting, and
Yeah.
I don't think there are too many who are following that right now.
Oh,
Well, perhaps in the future.
perhaps, yeah.
Okay, yeah, to Mark, to answer that question,
Right.
you know, I always do research and I put everything in a document, and then I go through what is interesting to pick and put forward. And one of the things is that you had a paper or topic, interfacing water and its potential role in the function of sericum against biofouling. And we had a podcast, it was also with Mark van Loosrecht, about biofouling and how microbial flora creates their own, let's say, habitat
Yeah.
by interaction of bacteria and secreting acids so that they feed each other on each other's outputs.
Yes.
And in a way, you wrote down like the water exclusion zone may facilitate anti-fouling behavior. Can you remember the one I'm putting forward, Jerry?
No.
The sericum is a globular protein whose resistance against fouling is important for application in biomaterials and water purification membranes. I have no idea what sericum is. You guys know that?
Well, I don't remember. When was this paper produced?
Oh, I have no idea. But on your website, I found news bulletins, and I picked one out, which was on biofouling. So I thought that was going to be interesting. Let's see if I can have when in contact with sericum, water molecules near the surface restructure to form a physical exclusion barrier that might prevent biofouling.
Ah, okay, silicon.
Oh, yeah, I mean, I can understand that. Since if easy water forms, it admits almost nothing into it, and so by forming that kind of a resisting surface, you can eventually prevent biofouling. I think that's what we were trying to say. Nothing more sophisticated than that.
No, but that would have huge impact. For instance, for the glass, the greeneries. We have lots of greenhouses here in the Netherlands, especially in the West. And
Yes.
one of the issues they have is biofouling of their water system. And this would I know some how do you call it, innovative greenhouse owners, and I'm gonna put this one forward to them just to see if we can trigger them to look differently at biofouling. They attack it with chemicals and everything.
Oh, that's a pity.
Yeah,
We don't want that.
And the way you describe it and the way you explain how easy is there and how it's working with infrared. I mean, infrared is also available in greenhouses. I mean, the light comes through, and maybe they could use that to do something good instead of attacking the bacteria.
Yeah, I mean, I think it's a matter of putting their minds to it, and I just want to say that we could do the same thing, but we're limited in what we can do. We're a research laboratory, and the research is oriented toward the most fundamentals rather than the practical applications. But those practical applications, there are so many of them, and it would be great to see interested people pay more attention to their development.
Yeah.
And in another one, you were mentioning that glyphosate is also bad for EC size.
What is bad? Sorry.
Glyphosate, 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 the weed killer.
Oh, glyphosate.
Yeah, sorry.
Oh, sorry. Oh, yeah. Well, I mean, so much evidence has been gathered, principally by, the person best known for gathering all of this evidence is my friend Stephanie Seneff from MIT. And the evidence that for the harmful nature of glyphosate is all over the place, and yet it's still being used. I'm not sure if it's used in Europe, but it's certainly used. It is used in Europe too. And we thought we investigated to see whether a possible mechanism for glyphosate's destructive nature could be in diminishing the amount of EZ water. As I said before, if you don't have enough EZ water in the cells, whether animal or cells, it's not going to function properly. It will be dysfunctional. And we did experiments, and we found indeed that that glyphosate diminishes EZ water in concentrations spanning the spectrum from concentrations that are used even smaller and smaller and minuscule concentrations. No matter what concentration you use, it will diminish EZ, except when you get down to vanishingly small concentrations when there's no effect. So yeah, this is it could be that this is the very mechanism through which how glyphosate works. And there are other poisons, and a question that we're intending to answer is whether those poisons may operate by this very same mechanism, by simply dehydrating. And I've been reluctant to ask the young students to pursue them because these compounds are dangerous, and so I'm waiting for a senior scientist to come along and say, yeah, I'm really interested, someone who knows how to handle these things properly. So we'll be addressing this in the future. We'll see.
Very, very interesting, Gerald.
Is there anything else, Gerald, you want to share? We're asking you a lot of questions.
Well, what I want to share is, well, I mean, I guess I want to share that people who have evidence against a theory, a prevailing theory, have a really hard time pursuing that, and we've done something to try to counter that. And I'll tell you in a moment. But the reason they have difficulty is, for example, if you have an idea that the Earth is round, and all your colleagues know that it's flat, they just look out the window, especially in Holland, where everything is flat, and and they say, well, just look outside. It's it's flat. We know it's flat, and you are saying, I looked at those satellite images and I see that the Earth has curvature to it. It's round, and please give me some money because I think the Earth is round, and if it's round and not flat, it has huge implications for everything. So please, I can't do anything without money. Give me some money. And you apply it to some organization, and the organization, the gatekeeper of the organization says, well, this looks pretty interesting. I should recruit the world's experts, the people who know the most about the shape of the Earth, to see whether these guys are crackpots, these Dutch guys, or whether there's something sensible. So who do they recruit? Well, they recruit the experts in flat Earth, the guys who study the little bumps that otherwise exist on the otherwise flat Earth, and they're going to review your application. And what do you think they're going to say? You're stepping on my toes.
Exactly.
And you won't get your money. They may be thoughtful and kind and say, well, it's a good idea, but your proposal doesn't elaborate on the necessary statistical analysis, et cetera, et cetera. You won't get your money. And so and all you get is a reputation for being a rebel that the establishment would like to get rid of. You see, so
officially in science, every theory should be falsifiable. Otherwise, it's not even a theory.
of course we all know that, but we're talking about human nature, and the system thwarts human nature. So we created something called the Institute for Venture Science. And that institute looks for ideas that challenge the status quo in cases where the status quo simply isn't working. It's just
exactly,
tradition that keeps it going. Wonderful. And we find money to fund these people, and we've identified 5 ideas, projects that if studies verify that they're right, they'll shake the earth in a major way. And we're looking for funding. We got a little bit of private funding from some people, and we're looking for people who have done well in their careers and want to give back in a meaningful way. And this is I think one of the most meaningful way, because these scientific revolutions that will be created will be very quick. This will happen in a matter of not hundreds of years, but in the next year or two. So we're looking for people who are interested, and all they need to do is contact me, and I'd be very happy to discuss with them these ideas.
It's a wonderful initiative, and you need some creativity there. Yeah.
Well, we need some creativity in helping to run and organize the organization, and also in identifying the most creative ideas. And sometimes the ideas are turn out to be bizarre nonsense, but other times they're really ideas with real promise, and those are the ones. And we do a very thorough vetting of ideas. We got more than 200 pre-proposals that were very thoroughly vetted, and from that we selected 15 for a full proposal and selected 5. So I think our review system is as thorough as any that you could imagine. And we'd like to be able to fund these, because they could lead to scientific revolutions that could then lead to the technologies that we need to solve our problems.
Exactly.
Yeah. Keep the water out of Holland.
And there is one more question I want to ask, because I forgot his name, Dwetsus, who was also inspired by your work, and they did the anode cathode experiment with a lot of voltage.
Yeah. Right. Okay. Tell me the question.
Because then, I mean, the experiment was that they had 2 Erlenmeyers with water. They put an anode cathode in, and suddenly there was a tube forming over the Erlenmeyers, and suddenly water was flowing from one Erlenmeyer to the other one.
That's Elmar Fuchs.
Yeah, that was the one, Elmar. And it was like a real bridge, and they could pull it apart like 1 or 2, maybe sometimes even 3 or 4 centimeters, and you could. I mean, it's like it was really, Mark. It was amazing. It was a real bridge in the air. Yeah. How is that connected to EZ?
Directly. It's EZ water that forms between those 2 electrodes, and you'll find that described in my 4th phase book. The 4th phase of water. I think Elmar doesn't agree with. I'm not sure with my interpretation, but the people doing the experiments have been struggling to understand what's going on, and they do indicate that there's some new phase of water. Whether they think it has anything to do with what we discovered, I'm still not so sure. But anyway, this is what I think is going on. Ordinary water doesn't do what you described.
Yeah, true. Yeah. It wants to go down. It doesn't want to go horizontal.
No.
Yeah.
It's an amazing observation. It's we have it working in our laboratory too. And when people come want to find out what's going on, we demonstrate. So it may be shocking to you.
Yeah.
But But yeah, it's really
Well,
a cool
just-
observation.
Cool.
Yeah, and it just reminds me. I don't know if that is true. I mean, I know here in the Netherlands they are experimenting a lot with plasma.
Yeah.
And it's also a certain stage of molecules, and in a way, it just I don't know. It reminds me a little bit of that, but I don't know if that is true.
I'm not sure either because it's really difficult to understand what goes on in plasmas. It seems to defy intuitive understanding.
Yeah, that's the feedback I'm getting, and that's why it's also a very interesting angle to solve a lot of issues we have.
Yeah.
Although it costs a lot of energy to make plasma, that's another issue.
Well, everything needs energy. And just one last point. I think you guys are about to sign off. Is I come back to where we get our energy, and you people may be aware that there are some people who go on without eating. Right. And so
some Indians,
True, yeah.
And it's possible. It's possible that it, without eating, it's hard to get ATP. On the other hand, if you're absorbing energy from the environment, then it's possible to build easy water. And the kinds of people who stop eating are very often the spiritually oriented people who may have a better capacity to receive this kind of subtle energy from the environment than the rest of us. So it's an interesting subject that remains to be studied, whether these people actually get their energy not from food because they don't eat food, but rather from energy from the environment, like infrared energy that we all get from the environment. It's an open question that remains to be studied.
There are also animals that don't eat for many years and still stay alive, like the olms in the caves. Like that. Very interesting stuff.
It's a question that needs to be answered.
We're going to discover who knows a lot about that, and then we just have another conversation with these persons.
Well, yeah. Why don't you? That's a great idea. Yeah.
Did you enjoy it, Jerry?
I enjoyed it very much. You guys are great. You guys are just so full of enthusiasm, open-mindedness, and a real delight. I'd like to extend great big hugs to both of you, but I'm not there. So the hugs will be by subtle energy for my bio field, and hope to meet you guys in person one of these days.
Yeah, if you ever visit the Netherlands, make sure to step by. Yeah.
Okay.
Or we just move? We just go to where you are in the Netherlands.
We can meet in the wintertime in Hawaii or somewhere where there's.
Wow. Love that.
Okay. That is a deal. Thank you for being on the show, Jerry.
My great pleasure. Thank you guys for your wonderful questions. Okay.
Thanks
Okay. Take care.
Take care.
We'll see you.
Bye-bye.
Bye-bye.
I hope you enjoyed this episode of the Master Beautiful Achievements podcast. A special thank you to our sponsors Scope Matters and Orvion for making this series possible. And do consider joining our Patreon community via the link here below. If you like our podcast, do subscribe to your favorite podcast app. And if you want to know more, you can find us on www.mastersofbeautifulachievements.com website. And if you like this podcast, do leave us a review on your preferred podcast platform. Then they know that they can rank this higher. Thank you for listening and join us the next time.